Hello, so this is a modified version of the talk given at the 2019 Geological Society of America meeting with co-authors Adrian Lamb and Nicholas J. Matsky. I am Sarah Sheffield, the person narrating this video, and this is a global biogeographical analysis of blastozoan echinoderms across the Great Ordovician Biodiversification Event, which I will refer to in this talk as the GOBI. So first, I'd like to acknowledge our funding sources and our institutional support for getting us to this conference and for being able to perform this research in the first place. So our story today, we'll introduce it by talking about the Cambrian explosion. So in the Cambrian, we have this first explosion of life that we really see in the fossil record because it's the explosion of biomineralized life, so things that are actually going to leave a fossil record. So in the Cambrian, we see a lot of groups evolving for the first time, and we see our higher order level diversity forming, so things like kingdom, phyla, classes, things like that. They all show up, but there's very few species within each of these classes. So if you look at these are all echinoderms, so related to our modern day starfish, sea urchins, etc. And there's very few species within each of these large groups existing in the Cambrian, 542 some odd million years ago. And there's also not a lot of ecosystem diversity, so things that are burrowing, things that are tearing, growing to different levels, they don't really exist yet. Everything's kind of doing about the same thing. But all of that will change once we cross into the Ordovician, into the Great Ordovician Biodiversification event. Again, I'm going to call this the GOBI so that I can actually say this without tripping over all the words. So what we see is a massive increase in the amount of lower level diversity. So things like species, genera, families, they diversify very quickly in geologic time, and they increase by about 4 times what they used to be. So here we have brachiopods, which have a really nice documented pattern here. So that's what you're looking at right now. We see across the Ordovician into the GOBI event, which crosses into the Darawillian here. It just explodes with diversity. And not only that, not only are we getting this high level of diversity with species, etc., we see a lot of ecosystem diversity development as well. So here we have some echinoderms showing off some of this beautiful ecosystem diversity. Here is Haplospheronus, and this has a holdfast, which would stick to hard grounds, possibly other organisms like brachiopods or bryozoans, kind of sticking onto that shell with a sticky mucus being emitted right here. Or we have Eumorphocystis with this lovely stem that would kind of stick into some soft sediment to anchor it a little bit better. So along with this biodiversification increase, it shouldn't surprise any of us that there were a lot of geochemical changes as well that are likely causing this biodiversification increase. So what we're looking at is a number of different isotope records. We have carbon-13 isotope records changing. We have large excursions, both positive and negative during this time. We have oxygen-18 isotopes telling us about the general warming and cooling trend that's happening throughout this. We're not entirely sure yet. Scientists are, of course, still working to uncover all of the geochemical reasons why this Great Ordovician Biodiversification event might have occurred in the first place. So hypotheses such as increases in oxygen have been proposed, etc. That's not really the focus of our talk today, but I wanted to introduce you to this a little bit. But the point is that with these these geochemical changes and the biodiversification increases, we should be able to look at the patterns that echinoderms themselves are taking to move throughout the world during this time. Few studies have been done to understand echinoderm biogeography, so moving through time and space, and most of the studies that have been done, the majority of these studies have been done without using a phylogenetic framework, meaning we're not taking into account the evolutionary relationships of echinoderms to be able to study this, which really means we don't fully understand these trends. We can't do any of these studies without that evolutionary analysis. Most of our analyses have been done using unnatural taxon grouping, so polyphyletic groups and paraphyletic groups as opposed to our monophyletic groups. And our main question here is, how did echinoderms respond biogeographically throughout the Great Ordovician Biodiversification event, the GOBI? So here we have one of the more recent biogeographic analyses of echinoderms from Lefebvre et al., 2013. And we see that echinoderms are changing their distribution. So the larger the dot, the more organisms that are found in a particular area. And they're clearly moving throughout the Ordovician, but we really don't know why. We can't track their patterns without understanding their evolutionary trends as well. So here we have a phylogenetic hypothesis of echinoderms. This is a fairly basic phylogenetic hypothesis presented in Severall and Ray 2007. We have our extant groups, for the most part, chilling out right here. We've got our crinoids over here. But today we're going to be focusing on this primarily extinct group, the blastozoans, that exist over on this portion of the tree. So our blastozoan echinoderms are those that are defined by specialized respiratory structures. We're not going to worry about those too much today, but each blastozoan group is defined by specific respiratory structure, meaning everything within that group should have that respiratory structure. So for example, the one we'll be talking about today are the diploporitans. Diploporitans are defined by having diplopore, otherwise known as double pore respiratory structures. So Eumorphocystis here again is showing off its beautiful respiratory structures. All these little holes here are those respiratory structures. So we have two pores connected by a single tube, and gas exchange presumably happened within those two holes. So part of the issue with these respiratory structures is that their morphologies are disparate. They're all very different from one another, even within the same group. So even though diploporitans have diplopores, many of them look very different from one another. And it's likely that there is a lot of convergent evolution happening within echinoderms. So our hypothesis is that maybe Diploporita, groups with diplopore respiratory structures, don't actually represent one group, and that they represent multiple groups, otherwise known as polyphyletic. So we did the first evolutionary analysis of diploporitans that has ever been done across all of the major groups of diploporitans. So there are 3, and those are highlighted here. We don't need to know their names to know or to understand what's going on, and we're going to take away these species names here and put them up with pictures instead. So what we see here is this red group, the spheronatids, make up one monophyletic group. They all grouped together in this evolutionary tree. However, the other two, astroblastids and glyptosphaeridids, show up across the rest of this tree here with non-diplopore bearing blastozoans. So we threw in a bunch of non-related groups to see how they would essentially, I'm going to start the sentence over, my apologies. We threw in a bunch of non-diplopore bearing blastozoans, so groups that are supposedly unrelated to diploporans, to test to see if they are in fact more closely related to diploporans than other diploporans themselves. And what we see is that diplopores themselves don't seem to be the reason, or don't seem to be linking this group at all together. And in fact, what we see is one hypothesis is that these respiratory structures have absolutely re-evolved through time. So we see that there's 2 diploporitan species over here with non-diplopore bearing groups entirely. So one hypothesis is that we have gained diplopores 3 times throughout the echinoderm evolutionary tree of life, and we've lost it once. This could have happened in many different ways. This hypothesis has not yet been tested. Research on it is underway. But what this indicates is that maybe there's some biogeographic reason why these diplopores are re-evolving through time. Why are they a successful way to breathe? So to start to test this idea, we defined 8 geographic basins. These basins are defined the same way as they are in Lamb et al. 2018. Here are our different basins, ranging from those in Laurentia, Baltica, and Gondwana, the major paleocontinents involved in this study. And we use this tree as our phylogenetic hypothesis. Now the tree on the next slide is going to look quite a bit different, but it is the same tree, same relationships. We have just time stratified the tree, meaning we use published locality information to tie stratigraphic dates to each of our species, and then we subjected it to tip dating methods and BEAST2 so that we have this beautiful fully bifurcating tree topology, excuse me. So our main focus today is on our Ordovician species and the speciation events within that group. And our question is, how did these blastozoans evolve across the Great Ordovician Biodiversification Event interval? So to do this, we used a program called BioGeoBears. Our package was developed by co-author Nicholas Matsky in 2013. And it's a biogeographic program that uses maximum likelihood to fit that uses models fit to the data itself. So here are some examples of previous studies that have used this. There haven't been too terribly many yet, though they are starting to be published at a higher rate. We have most of our organisms being used in these techniques and being brachiopods and trilobites, very common invertebrate taxa at the time. The best thing about BioGeoBears is it's one of the only biogeographic programs that allows for only fossil data. So it's an excellent chance to test our paleontological hypotheses of biogeography. It has never before been used in echinoderms, so this is an excellent opportunity to start to look at how echinoderms have moved through time and space here. So the model for our particular study is highlighted here in yellow, the one that we're going to be using. So these numbers don't need to mean anything to us right now, but essentially the model that best explains our data is the one that has the lowest AIC score, and that would be here, the DIVA-like plus J. So dispersal and vicariance analysis, the plus J indicates a high degree of jump dispersal within the tree. So vicariance dispersal here, the two modes here, vicariance is when we have a barrier separating the two species, and then we have dispersal, which is happening a little bit differently. We have a small population over here, and they eventually develop into two without the reason for that population developing solely being that that geographic barrier. So what we see is we have a Gondwanan origin for our Ordovician clades. So all of our Cambrian organisms here are from Gondwana, and we have strong affinities between Baltica and Cincinnati regions, as well as Gondwana and Laurentia. Now this does have a sampling bias, as Gondwana is not as well sampled as other paleocontinents, but this is something that we'll discuss in a little bit. We also see that it's dominated by dispersal events throughout the Ordovician. We have our dispersal events being highlighted by a yellow triangle and our vicariance by a red circle. All of our vicariance events happen in the Cambrian, so right outside the scope of the image that we're looking at. So here we have a pattern of the dispersal patterns of blastozoans. We have the red lines indicating the Darwinian age events that is within the Gobi interval there. And what we're looking at is most of our organisms seem to be using volcanic island arcs, so shallow carbonate platforms, to get across large ocean basins like the Iapetus Ocean. So how this is working is that echinoderms have a larval stage, even those that are sedentary or sessile, meaning stuck to the sediment or something like that, or stuck in a particular place for the rest of their lives. They have a larval stage that they actually use to move around and go into different places. So we're seeing that most of these organisms are actually taking long pathways across us, indicating they're likely using shallow carbonate platforms, volcanic island arcs, which are fairly common at this time due to the high level of tectonic activity. So to look at our paleoclimatic influences, we can highlight the Gobi interval itself, again in the Darwinian, and we again see that blastozoans here, the diploporans and other groups, are dominated by dispersal events. But we can actually compare this to other invertebrate taxa at this time with published data. So we have brachiopod speciation and trilobite speciation, so this is compared to Adrian Lamb et al. 2018's paper. We see that all three of these groups are really dominated by dispersal events. And we see an interesting pattern here of when we have total organic carbon mercury signatures coming in, which can indicate large igneous province or volcanic activity, we see that blastozoans and trilobite speciation really stops, whereas brachiopod speciation really doesn't seem to be affected by that. So this is a pattern that we don't necessarily have a whole lot of information about yet, but it is something interesting to note. So a couple of notes about this is that Baltica and Laurentia remain the best sample paleocontinents for Ordovician echinoderms. Other areas of the globe, for example, South China, Gondwana, etc., Perigondwana, remain undersampled. So increased sampling within these undersampled parts of the world will certainly provide additional data and strengthen our interpretations. So to conclude, this study represents one of the first biogeographic analyses of echinoderms based in phylogenetic hypotheses. And evolution of these echinoderms is dominated by dispersal events throughout the Ordovician intervals. The Gobi interval is characterized by strong affinities between the Cincinnati Basin and Baltica, as well as Laurentia and Gondwana. And these echinoderms seem to follow similar biogeographic speciation patterns of other invertebrate taxa at the time, for example, the trilobites and the brachiopods. And with that, I thank you for listening.